I welcome you all and thank you especially for accepting this invitation.
Thank you, ma'am. My pleasure, ma'am.
Yes. So I think Usha, the student जो भी है, we can start now lecture. So I can just introduce Ankit. So as we all know, today we are having this, the lecture on the sweet science, eminence of O glycan in hematopoiesis. The three types of blood cells, RBC, WBC, and platelets, arise from hematopoietic stem, progenitor cell in the adult bone marrow, and function in physiological regulation and communication with local microenvironment to maintain the systematic homeostasis. Major glycans are N-glycans, O-glycans, glycosaminoglycans, and glycosphingolipids. Aberrant glycan expression occurs in most hematological malignancy, notably acute myeloid leukemia, myeloproliferative neoplasm, and multiple myeloma. Today we have the speaker, Dr. Ankit Tanwar, who is the alumni of the Department of Biochemistry, Shivaji College, batch 2009 to 2012. He did his master's degree in toxicology from Jamia Hamdard in 2014, followed by PhD in toxicology from Institute of Nuclear Medicine and Allied Sciences in Mars, Defense Research and Development Organization, DRDO, Ministry of Defense, New Delhi, Government of India, in collaboration with Jamia Hamdard, New Delhi. Dr. Ankit Tanwar is currently working as a postdoctoral scientist in Department of Cell Biology at Albert Einstein College of Medicine, New York, USA. He's working on NIH project entitled Role of Glycosylation in Notch Signaling. He also received Young Investigator Award for the development of novel animal model in the field of infectious rheumatology from International Conference on Controversies in Rheumatology and Autoimmunity, held in March 2017 in Bologna, Italy. He is a key worker of team who coined the word herbal informatics to derive drug molecule from the natural products and activity, working to provide the curative remedy for the disease of unknown etiology. Recently, his model, herbal informatics, has been used for the initial screening of emergency approved drug by DCGI 2-deoxy-D-glucose for combating COVID-19 situation in India. Dr. Tanwar has published more than 20 research articles in peer-reviewed journal and participated as an author in 2 books titled Demystifying Escape Pathogen and Ebola Virus. He has also received a various prestigious awards from ICMR, DST, Jamia Hamdard for their excellence in research. He is also serving as an editorial board and co-chair of Reputed Journal Organization Conference Award Committee and the member of organizations such as American Society of Hematology, Society of Toxicology USA, Society of Life Sciences USA, Bentham Service Ambassador USA, and also actively participates to deliver invited talks at international conferences. Dr. Tanwar has expertise in the various areas of life sciences, including toxicology, cell biology, hematopoiesis, immunology, rheumatology, glycobiology, and herbal medicine. I am very much sure that today this lecture will enlighten us our knowledge and specify to the important major points where we can continue and understand what is the role of glycans in hematopoiesis. So I would request Dr. Ankit to please take over from this and continue your talk about this glycans.
Thank you so much, ma'am, for your very nice introduction. Actually, it is a privilege for me to be here and deliver a lecture in front of my alma mater. And okay, so it's shared, right? You can see the screen.
Yes, we can see your screen.
First of all, I would like to express my gratitude to all the staff of Biochemistry department. And so today we will talk about the sweet science, eminence of O glycans in hematopoiesis. You can see this is a very beautiful view of Albert Einstein College of Medicine campus. Long tall buildings and different labs. So like starting from the first of all, I would like to brief the outline of the today's talk. So I will be talking about the hematopoiesis, just a brief introduction for all the students, the glycans, what they are, where they are, why and how we should study glycans. Then I will be introducing the use of animal models. We have developed several compound mutants to study the glycans and their role in the hematopoiesis. And then I will introduce one very advanced flow cytometry technique that is recently launched in 2018, Cytec Aurora. And then O glycans in hematopoiesis. And after all that, I will be introducing the herbal informatic that is very a popular tool for the drug discovery. So starting from the introduction of hematopoiesis, as you all of know, you must have heard that hematopoiesis is the process for the generation of new blood cells, and they produce the blood cellular components, and all the blood cellular components are derived from the hematopoietic stem cells. In adults, we have trillions of cells produced daily, and these hematopoietic stem cells are generally resides in the medulla of the bone. They are unique because they have self-renewal capacity and they has ability to differentiate in the any cell types and tissue. So okay. Now if I talk about the bone marrow, they are found in the spongy gelatinous tissue found in the hollow spaces in the interior of bones. You can see in this figure. And then in children, they mainly occur in the long bones, that is femur and tibia. And in adults, they are occur in the pelvis, cranium, vertebra and sternum. This is very important word actually, extramedullary hematopoiesis. That is the main site for this hematopoiesis is liver and spleen. When the hematopoietic stem cell system is disturbed in the body, liver and spleen has potential to generate the blood cell types. So you can see the bone marrow, and then you can see here we have different cell types, that is stromal cells, blood vessels, bone matrix, and how this hematopoietic stem cell differentiate into the different cell types I will be discussing later. So let's move towards the next slide. Bone marrow contains two types of stem cells, mesenchymal stem cells and the hematopoietic stem cells. Red bone marrow, we have two types of bone marrow, red bone marrow and yellow bone marrow. Red bone marrow is generally containing the hematopoietic stem cells. They are blood forming stem cells. And yellow bone marrow contains the mesenchymal stem cells that are usually produce fat, cartilage and bone marrow. Stem cells are immature and then they can turn into a number of different cell types. Hematopoietic stem cells has two major lineage cells, that is myeloid and lymphoid. And we call this as the army of our body, army for the immune system. And mesenchymal stem cells are present in the bone marrow cavity. They differentiate into the number of stromal lineage cells, that is chondrocytes, osteoclasts, adipocytes, myocytes and macrophages. And it is important to learn that before birth, but towards the end of the fetal development, these stem cells are developed, and they first develops in the clavicle and becomes active about 3 weeks later. Bone marrow take over from the liver as the major hematopoietic organ at the 32 to 36 weeks gestation period. And the bone marrow that is red becomes red and remains red, until the age of 7 years, as the body required the production of more blood cells during that period. As body ages, it gradually replaces the red bone marrow with the yellow bone fat tissue. And in adult, it has been seen that we have 2.6 kilogram of bone marrow, and about of which half is red. Their highest concentration is mainly occur in the bone of the vertebrae, hips, breastbone, ribs and skull, and so on. Now, let's discuss about the warriors. Why I'm discussing the basic overview of this hematopoiesis is because we are using different knockout of glycans and we are studying these warriors. So the warrior of the immune system are lineage cells, which I have mentioned before, lymphoid and myeloid mainly. In lymphoid, we have T cells, B cells, NK cells, this is natural killer T cells. And we have other part like immunity, blood clotting factors, red blood cells, and erythrocytes that carry oxygen. Okay. There is some problem, I think. Okay.
क्या हुआ अंकित?
मैम एक्चुअली वो स्लाइड स्टक हो गई थी बीच में, but now it's okay. Then if I talk about the lymphoid cells, they are differentiated from the pluripotent stem cells. They are actually white blood cells and they play a very important role in our body. They protect the body and defend the body against the cancer, pathogen and foreign matters. In the lymphoid cells, we have different cell types, T cells, B cells and natural killer T cells. B cells are important for the production of antibodies in response to the T helper and T cytotoxic cells, and T cells generally destroy the body own cells that are infected with any foreign material. Natural killer T cells are generally the heterogeneous population and they actually provide the bridge between the innate and adaptive immunity. These cells, lymphoid cells, are majorly circulate in the blood, lymph fluid, and they also found in the body tissue such as spleen, thymus, bone marrow, and lymph nodes. If I talk about the B cells and T cells, B cells are important for the production of antibodies. That's why they are called as a antibody mediated system. And these antibodies are mainly produced in the spleen or lymph nodes. T cells are mature in the thymus. They have the cell mediated immunity as well as the adaptive immunity. And there are major two cell types which play a very important role to protect the body from the foreign material, T helper cell and T cytotoxic T cells, which actually when there is any class 1 or class 2 MSC molecules they found and they have an antigen presenting cells, then they activate the B cell as well as they activate the cytotoxic T cells also, which produce the pro-inflammatory cytokines, and the B cells produce the antibody in response to the cytokines level. Okay. And if I talk about the myeloid cells, this is the primary responder of our body army. Like they are the primary responders, which actually associated with the innate immune system, inborn or natural. They are granulocytes, monocytes that are differentiated from the pluripotent stem cells. So we have different soldiers here in our immune system that is called as macrophages, neutrophils, basophils, eosinophils, erythrocytes, dendritic cells, and platelets, and they have their own like potential and own function to play and to protect the body from the foreign material. And myeloid cells play very important role in the immunity because they are the first responder when any foreign agent attacks in our body. And after that, they activate the secondary responders that are the lymphoid lineage cells, and they like together fight the foreign material. Now, what is innate and adaptive immunity is? So innate immunity is fast response. They generally occur after the attack of pathogen. They activated within the 4 hours, and we have different cells here which play a very important role in the innate immunity, that are natural killer cells, mast cells, basophils, eosinophils, neutrophils, and the complement proteins. And in the adaptive immunity, we have slow response because after the attack of pathogen, innate immunity activated, and then they further activate the other pathways which will give to the adaptive immunity. And adaptive immunity has major two response, that is humoral, in which the B cells produce antibodies, and we have cellular immunity in which T cells majorly play a very important role to activate the immune system. So this is the overview of how lymphoid and myeloid cells are important for the body. and they play a very important role in the immune system. So in the niche bone marrow cells, we have multipotent hematopoietic stem cells. This is just the overview of how they have differentiated into the common myeloid progenitor cells and common lymphoid progenitor cells. I have already talked about myeloid progenitor cells are the part of innate immunity. They are the first responders. So we have here thrombocytes, erythrocytes, mast cells, myoblasts, which further secrete into the different cell types, basophils, neutrophils, eosinophils, monocytes, and they further produce the macrophages. In the common lymphoid progenitor cells, we have two main cells, lymphocytes and the natural killer T cells. In the lymphocytes, small lymphocytes, they are further differentiated into the T cells and B cells, and so B cells has function to produce antibody in response to the cytokines, and T cells generally produce the cytokines to kill the target, to eliminate the foreign antigen from our body. So we have other immunoglobulins that we know that we have 5 classes of immunoglobulins, and we have different function of that like IgG has bacterial required for the most abundant antibody actually, and these antibodies are required for the bacterial and viral infection. This is the structure of different antibodies. We have secretory antibody that is present in the body surface, mucous membrane. IgM is the antibody which is produced, like first antibody that is produced to fight against the infection. Then we have hypersensitivity reaction in our body, and we have IgE that is required for the allergic reaction. IgD, the role of IgD is still not fully understood, because it is like most of the scientists use this IgD antibody to discriminate the different B cells, but like pro B cells, pre B cells. Okay, so this is all about the hematopoiesis, just a general overview so that you know what actually hematopoiesis is and how many cells we have and how they are actually reacting and acting in our body. Then let's discuss about the glycans. Why glycans are important? So we know that glycans are chain-like structure that are composed of the single sugar molecules, monosaccharides, linked by the covalent bonds. Monosaccharides such as fructose, glucose, galactose are widely known as building blocks of dietary sugars and carbohydrates. They are actually the main source of energy in our body. They are the fuel in most living organisms, and they are branched like in a way in which they can produce so they actually linked with the different sugars, monosaccharides, and they branch in a way in which they attach to the surface of the protein, and where they play a very important role in the immune system. Chains are not only a source of energy, they also have many biological functions, which we will be looking for, how they are actually responsible for the immune cells, how they are responsible for the differentiating the different blood cell types and all, while they are making the glycoconjugates. So this is actually the main figure in which you can see the glycan structure, which is attached with the different glycans, sialic acid, sugar, galactose, N-acetyl glucosamine, mannose, and fucose, and this is just the representative figure of these structures. So when I was studying this, whenever I see color, why this pink is sialic acid, why not it is galactose? So when I arrived here in my meeting, I asked with my boss why it is not yellow, why we are calling this galactose. Then my manager told me, and I just asked who decide actually these colors, and just then I got to know that my boss is the core committee member who decides this color actually. So here we can see the pink color is, this pink is denoting the sialic acid, yellow one is the galactose, and this is the N-acetyl glucosamine, and which we have mannose attached with it. We have another N-acetyl glucosamine, and we have fucose here. So these glycobiology is actually a very rapidly growing field, and these glycobiology are using actually for the drug discovery also, because they have a very different role, which is not actually fully determined yet. Then sugar chains known as glycan, we know they are found throughout the human body, attached to our cells, many proteins, antibody, and also to some viruses and bacteria. They are very important for the biological process, and they actually used for the communication and identification of various markers also. That's why it is called as a third chain of life also, after the nucleotides and peptides protein. The interest in the use of glycans are okay. The interest in the use of glycans in healthcare and drug discovery are rapidly growing because the scientists are working very much in these glycans, and they are revealing the new identification of these glycans nowadays. Several ways in which the biological role of glycans can be harnessed to develop new glycan-based drug and treatment. This is actually the advanced tool nowadays scientists are using and making the glycan-based drugs and treatment. So if we look here, we have how we can use glycans and for the treatment and the drug-based glycans. So glycans can be used to fighting the infection, because disturbing ability of bacteria and viruses to bind with specific glycans on cell surface could be used to prevent the infection like influenza that targets certain tissue and organs. So actually influenza virus use the surface glycans, and then they endocytose towards the host cells, and where they did the fusion and uncoating. Also glycans can be used they can also be used to reduce the antigenicity, because when we administer any pharmaceuticals drug without glycans, sometimes body reject it. So the drug which are attached with the glycans then can also reduce the antigenicity, and body will not reject it because they will found as a self glycan also, because the glycans are highly decorated with the antibody surface also. Now they can also use as to prolong the therapeutic effects with and without glycans. They have other role also. They can be used to directly, if we use glycan in the drug, we can use it as a direct target drug. We can directly deliver the drug to the target cells, where they can play an important role to kill the cells. Also, the very important, improving the drug solubility. If we use the glycans with the drug, they increase the solubility in water, which making them easier to handle and administer to patient, and it definitely improves the absorption also. Then why I'm discussing about glycans in a cell, how they look like. So you can see the microscopic view of the cells, and if you enlarge this microscopic view, you can see these are the cells, and these hair like structure are all glycans. They are different glycans are attached to it. And if you'll see the 3D image of these, so you can see the cell surface, and we have the glycoprotein in which the glycans are beautifully attached with the protein, and you can see the protein and glycans over here. So how they are attached with the surface, and they can be used as a marker of different cells. Now cell surface glycan act as a marker for cell identification, which I have just mentioned. Glycans are responsible for the existence of ABO blood grouping system also. You can see different blood types: group O, group A, group B, and group AB. You can see we have here the erythrocytes, which are attached with the galactose. Then we have a glucanac, that is N-acetylglucosamine. Then we have a galactose, and then fucose. But in the group A, we have one extra N-acetylgalactosamine, which are attached with the galactose. And in the same manner, we have here two galactose that are attached together. And in the AB blood grouping system, we have like branched glycans attached to the surface. So glycoproteins are not only found with the cell surface, they can found inside the cell and also the extracellular matrix of the protein, and which makes them eligible to study in the signaling. So we can use these extracellular metrics to study the different signaling pathways and to see how these glycans can affect the particular pathway. And if we regulate that pathway on the basis of these glycans, it might help for the treatment or just to find out the mechanism, what's going on inside the cells. And this is the one overview of how influenza virus binds to the glycansialic acid molecules on the surface of host cells, and then the endocytosis, and then further the other steps, fusion and coating happen. Now I will be discussing about the crosstalk between the virus and the cells. So this is, you can see how beautiful the immune cell is, which is highly decorated with the different lollipops, which are glycans. And these viruses are looking for these glycans that look like a lollipop for them. They look and they're saying, look, I found a sweet spot, and they are asking their neighbor virus also, okay, come behind me and target them. And here the immune cell are asking, hey, you look sweet and tasty to the virus. And this virus also have a glycans, which is actually recognized by these immune cells. But this virus no, and they are saying, who, me? No, I'm sour, not sweet, I'm just like you. So how beautifully they interact with each other, and like immune cells has potential to recognize whether these are self glycans or foreign glycans. If they found foreign as self or the self as foreign, they can cause the autoimmune disease. And that's why glycans are important to study, because these sugar codes are very difficult to study. And now lots of research in USA, they are looking for to make these maps so that they can easily identify the different cell types, different glycans on the surface of different cells of virus as well as the human body. And just to show how these healthy cells are different from the tumor cells or affected cells. So we have normal glycosylation here, and you can see here glycans are attached on the surface of the cells, and they are actually branched and well mannered. So that's happened with the different enzymes that are responsible for binding, catalyzing the addition of these different glycans on the cell surface. But if you see the tumor cells, we have apparent glycosylation because it is not well branched. It is like, you can see the surface is heavily decorated with the glycans, and this is actually the indication to identify how the healthy cells, normal cells, can be differentiated from the tumor cells. Glycans are important for the cell-cell interaction and communication because they can control the behavior of the immune system. Glycans also has one of the indicator to target whether this is the self-glycan or the foreign glycan, because if we know the sequence, we know the sugar code, we can easily identify whether it is self or foreign. Also, glycan are essential component of antibodies which binds to the pathogen and infected them. Either they neutralize them and or they tag them for the removal by the WBCs. Incorrect glycan structure, which I have just mentioned before, that if glycan structure recognize self as a foreign, they can start killing them, and which is responsible for causing the autoimmune disease. So nowadays, many pathways like no signaling and these glycans are studying in the autoimmune disease also. Now, okay. Now, I will be discussing majorly on the O glycans, but I would like to mention about the N glycans also. These glycans are majorly found on the exterior surface of the cells. They both are present in eukaryotes and prokaryotes. N-linked glycans are attached in the endoplasmic reticulum to the nitrogen inside chain of asparagine, and it is a sequence actually. So they bind on the in between the asparagine and the serine, or asparagine or threonine. In eukaryotes, all linked glycans generally bind to the hydroxyl group of the serine or threonine residues in the ER, Golgi, cytosol or nucleus. However, these N-linked glycans majorly in the endoplasmic reticulum. Why O-linked glycans are important? Because O-linked glycans are important for the hematopoiesis process. it's required for the inflammation response mechanism also, and they are responsible for the APO blood antigen also. However, the N-linked glycans also has their own function and role. And if we talk about the O-linked glycans, elongation and termination of these O-linked glycans is carried out by the several glycosyl transferases, which I will discussing about 3 glycosyl transferases that are actually very important for this elongation and termination. Okay. And now notch signaling. I just mentioned before that these glycans are also controlling the role of different signaling. So notch signaling is a very important signaling pathway that is required for the cell fate decision and determination. In mammals, there are 4 different notch receptors, starting from the notch 1 to notch 4. However, Drosophila has only one homologous, which is more similar to the notch 1. What is notch? And we have here notch receptor and notch ligand. Notch receptors are signal transmembrane glycoprotein comprising of notch extracellular domain, transmembrane region, and an intercellular domain. And in the signal sending cells, we have different ligands, majorly 5 ligands in the mammals: delta 1, delta 3, delta 4, jagged 1, and jagged 2. And this notch extracellular domain is consisting of 29 to 36 EGF repeat, and each repeat is consisting of 40 amino acids. And we can look, okay, let's first study the how notch signaling works, then I will be moving towards the extracellular domain of notch 1. So notch signaling, this is the one cell that is expressing the notch ligand, delta or jagged. And when the notch spans the cell membrane, it ligand binds to the notch receptor, which is expressing by the different cells, and it induces the proteolytic cleavage. The first cleavage caused by the metalloproteases, that is ADAM10, and the second cleavage, and then these like ligand which is bind to the extracellular domain, endocytose, and the second cleavage is caused by the gamma secretase, which cleaves the intercellular domain that are attached with the extracellular domain, and this intercellular domain releases toward the site of nucleus, where it make a complex with the RPPJ, very important for the notch signaling. It's the transcription factor, and this complex recruits the other co-activator mastermind like, and they activate the notch signaling by activating the different downstream genes. Now, why I am discussing about notch signaling? Because I was discussing about the O-glycans. O-glycans are highly decorated in the notch extracellular domain. And if you look at particular this point, you can see this is the mouse notch 1 extracellular domain, and here I will be discussing about the eminence of glycans in hematopoiesis. So this is mouse notch 1 extracellular domain that is consisting of 36 EGF repeat, and this each box is showing the one EGF that is consisting of 40 amino acids, and that is further comprises by the 6 cysteine residues, and in between the cysteine residues we have serine and threonine in which the glycans bind. So you can see O-fucose glycan, because I am discussing about the O-glycans, so they are all O-fucose, O-glucanac, and other. So POFUT1, it's an enzyme that is protein O-fucosyl transferase 1. And what is it? It actually catalyzes the addition of O-fucose in the EGF of notch protein, and this repeat is further elongated by the fringes, that is lunatic, manic, and radical fringes. We have another enzyme, EGF repeat O-glucanac transferases, that catalyze the addition of O-glucanac in between the 5th and 6th residues, cysteine residues, and they attach in the serine residues. So these are actually very important enzymes to study the notch signaling. But deletion of this gene causing the different genetic disease, that is Dowling-Dego's disease. Deletion of these fringes, that is mainly by the lunatic fringe, causes the Spondylocostal Dystrosis, and the EOGT causes the Adams-Oliver syndrome. Let's look what like how and what they are. So if you see the downlink Dagos disease, it is characterized by the hyperpigmentation of the skin, abnormally dark skin coloring. It is a very rare condition, and so far the case reported in the literature is 50 only. But nowadays they are increasing also, because the hyperpigmentation is the like main problem nowadays, and it is very difficult to discriminate whether it is causing by the ultraviolet or these genetic mutations. Mutation in the keratin 5, POFUT1 and POGLUT1 gene is responsible for causing the Dowling-Dego's disease. And this condition is inherited in an autosomal dominant pattern, and this Dowling-Dego's disease has no racial or gender predilection, and lesions typically present in 3rd to 4th decade of life. So you can see the images of like from the literature in which you can see the hyperpigmentation of skin. Here also they have defined the spot and they measure the spot, and this is the histopathology they have shown. Then Adams-Oliver syndrome is caused by the mutation in the DLL4 EOGT that I have just discussed about the EOGT enzyme, Notch1 and RPPJ gene also. It is also a rare condition that is characterized by the congenital disorder. It is first described by the Adams and Oliver in 1945. The primary feature is malformation of limbs and the abnormalities in skin development. And also you can see this skin pattern of this baby, and this is the James, 3-year-old boy that is suffering from the Adams-Oliver syndrome. He is in USA, and their inheritance pattern is decided by the which gene is mutated actually. If it is caused by the mutation in the DLL4, that is delta like 4 gene, Notch1 and RPPJ gene, it is the autosomal dominant pattern, and EOGT gene is generally the autosomal recessive pattern. And so far 125 affected individuals have been reported in medical literature. And this is the another disease, genetic disease, that is Spondylocostal Dystrosis, that is characterized by the abnormal development of bones in the spine and ribs. You can see this image of human beings, how like they look like, and majorly it is caused by the DLL3 gene, but lunatic fringe gene is also causing the severe spondylocostal dystosis. I will show you the animal model of where we have knocked out this lunatic fringe gene and how the mice model looks like when we compare with the control mice. They also depends on both the autosomal recessive pattern and the autosomal dominant pattern. Let's discuss about the notch signaling in the immune cell development, because our main goal is to study the glycans in the immune cell development. So I have already discussed about the lymphoid and myeloid lineages are the main soldiers in our immune system, which play a very important role to protect our body from the attack of foreign antigen. So hematopoietic stem cells in the niche hematopoietic stem cell, they are differentiated into the multipotent progenitor cells, which they have different types of multipotent progenitor cells, starting from the multipotent progenitor 2, multipotent progenitor 3, and multipotent progenitor 4. Then these multipotent progenitor cells further differentiate into the common myeloid progenitor cells and common lymphoid progenitor cells. And these lymphoid progenitor cells arises in the thymus, where they have won the different stages for the development of T cells, which you can see here, early thymic progenitor cells, and they have different double negative stages for the development of T cells. And you can see here thymic epithelial cell, when they arises in the thymus, they actually delta like ligand 4 that is required to induce the Notch1 signaling. And Notch1 signaling is very important for the development of these three stages, different stages for the development of T cells, which you can see early thymic progenitor cells, then they move towards the double negative stages, and then they finally differentiate into the single positive CD4 T cells and single positive CD8 T cells. However, thymus has also potential to produce the B cells, if the Notch1 signaling is disturbed, the early thymic progenitor cells that they just arise in thymus from the bone marrow, and the double negative 2 stages cells has potential to secrete the B cells and myeloid cells in the thymus, and the production of these cells directly indicate the disturbance of Notch signaling. However, Notch1 signaling must be switched off for the development of B cells, and here you can see the Notch2 signaling that is induced by the delta-like ligand 1, and the RPPG kappa is required for the development of B cells as well as the dendritic cells. And you can see here the different transition stages from starting from the T1 to T2, and then they differentiated into follicular B cells and marginal B cells. Still, it is not clear whether this Notch2 signaling is also required for the follicular B cells development or not, and I am studying to find out whether here is also the Notch2 signaling is responsible. Now, mice model which we have used to study. So as I have mentioned that Notch1 extracellular domain of mice is highly decorated with the different glycans, and this different enzyme, that is EGF, EOGT, POFUT1 and fringes, are required for the addition of different glycans in the EGF of Notch protein. So we have actually different glycans knockout, which my lab developed a few years back, and I'm crossing it to develop the different conditional mutation and the global knockouts. So what we are studying actually, so this is EOGT that is adding, I have just mentioned the EOGT is required for the addition of O-glucanac in the EGF of Notch protein. So you can see the O-glucanac here, this blue boxes. So we are studying, are the EOGT or O-glucanac glycans required for the immune cell development? We have developed then the EOGT knockout mice model. Then we are also interested whether, because the EOGT is also adding the O-glucanac and the fringe is also adding the glucanac after the O-fucose is added by the POFUT1. So if we remove the fringes, totally the glucanac from the surface of EGF surface, then what will happen? How it affect the Notch signaling and how it affect the immune cell development? So we developed these triple knockout. The fringes are radical, manic, and lunatic. So we knock out these fringes and the EOGT enzyme. Also, we were interested to study the whether this, if we knock out POFUT1 and EOGT together, like then the surface of the nose don't have O-fucose and not the O-glucanac. So what will happen? Whether do O-glucanac glycans support the Notch signaling in mice lacking O-fucose glycan? So here we have developed the conditional mutation of POFUT1 in hematopoietic stem cells. And why we have developed the conditional mutation in the hematopoietic stem cells? Because the global knockout of this POFUT gene is embryonic lethal. Now this is the mice model which we are using and to study the different immune cells. Now I have mentioned in my outline that flow cytometry is a very advanced techniques. You must have heard about the BDLSR and BDLSR for TESA. But Cytec Aurora recently developed a very famous technique of the flow cytometry, which is nowadays very popular in USA, and I don't think it is launched yet in India. It is 5 laser and 64 fluorescent detector, and the beauty of this machine is it can detect, if you have your blood sample or any sample, it can detect the 40 markers together in a one experiment. You don't need to make 1000 tubes for the 1000 markers. So you can, in one tube, you can identify the 40 markers. You can study the immune system very quickly. This is the basic protocol which this machine, actually the flow cytometry we are using. We use the single cells or the peripheral blood. We use the FC block, that is antibody that block the FC receptor, so that our antibody which we are adding to identify the antigen can directly hit the target. And then after the incubation with the FC block, we can use the fluorescent antibodies and incubate the cells with this antibody, and then we further wash the cells, and then we studied in the different markers. And like for all the students, I would like to say, if you want to go for the research scientist job after your masters, this is the very good tool to learn about, because maybe soon this machine will be there in India, and most of the company will be hiring the staff or scientists, junior scientists, senior scientists to handle this sophisticated instrument, because it has multiple colors and it takes like comparison time a lot. This flow cytometry is very popular in the different fields like cancer research, immuno-oncology, immunophenotyping, hematology, and various quality control as per the SOPs set by the companies. And this is just a representative overview. I'm not going to give the details of it. So this is how the representation of the flow cytometry looks like. Here we have used the thymocytes marker. I just mentioned that we can use multiple markers in that machine. So this is just an example. We use these 5-6 markers, and this is the fluorochrome which is attached with the different antibodies, and that helps actually flow cytometry to identify these particular markers in the cells, and we can study like how these cells can be used. Okay, so I was just talking about, we have developed the mouse mutant. So here you can see that does EOGD and O-gluconate glycans affect the hematopoiesis? So we have EOGT enzyme, O-gluconate transferases, and we cross these mice with the EOGT null mice. So you can see this is the one EGF part where these O-gluconate is attached in the EGF of notch protein, and also the fucose and gluconate is there. But when we knock out the EOGT, because EOGT is the enzyme that is required for the addition of O-gluconate, so we remove the O-gluconate here. We have only O-fucose and gluconate that is added by the fringe. You can see how now the mouse notch 1 extracellular domain looks like. So you can see the O-gluconate is here because this is the control mice. But when we remove the O-gluconate, there is only O-fucose here. So what we studied, this is actually the unpublished work, so I'm not going to show you the data. So, and when we studied these mice, so we found that it is required for the lymphopoiesis and myelopoiesis highly, and it is not dispensable actually. So actually our lab and one lab in the Japan has competition to who will publish the first about this model. So our manuscript is all ready to communicate. That's why I'm not showing the data here. And also we were interested whether these EOGT null mice samples has cell intrinsic property or not. So what we have done here, we did the bone marrow transplantation study, and in the bone marrow transplantation study, we used the cells, bone marrow cells from these different control and the mutant cells mice, and we injected retroorbitally. This is the retroorbital plexus route. We can inject and collect the blood from the plexus of the eye, and we actually injected using this route in the recipient mice, that is plex 6 congenic strain. And after the 7 weeks of the transplantation, we studied these different markers, and we found that these cells has autonomous property, and they have cell intrinsic function also to some extent. Now if you look, superfood is also required for the addition of O-fucose, and so this is the one of the model which we used. Actually this is a published paper in the collaboration with our lab. So you can see here the control is there. It's same as we used in the previous model, but in the POFUT1 enzyme we don't have glucose here. Oh sorry, O-fucose here. So you can see the mouse extracellular domain, there is no O-fucose, right? And here we have, I mentioned before also that POFUT global knockout is embryonic lethal. So we use the MX3 mice model, to inactivate the immune hematopoietic stem cells, to activate this POFUT enzyme in the hematopoietic stem cells. And when we studied these cells, different spleen cells, thymus cells in the flow cytometry, we found neutrophils is increasing in comparison with the control. This is the different representation of the bone marrow cells, where you can see this neutrophil, GR1 is a marker for the neutrophils, is increasing, and also here it is increasing. This is very important, as I have discussed in my first or second slide, extramedullary hematopoiesis, and I have mentioned that hematopoietic stem cells, when disturbed, they ask the spleen to generate the blood cells. So here, because we have inactivated the POFUT enzyme in the hematopoietic stem cells, and that is directly affecting the hematopoietic system, and we found that splenomegaly occur in the spleen, and that is, and then we found in the histopathology analysis also, like in my study, I have found the clear infiltration of this extramedullary hematopoiesis, and this splenomegaly is actually indication of the extramedullary hematopoiesis that is caused by the disruption of hematopoietic stem cells. Okay, and then we also found, because the hematopoietic stem cells are disturbed here because we knock out the POFUT1 enzyme, and the T cells are also arising from the bone marrow. So we found thymus is very tiny in comparison with the control, because there is no T cells developed. T cell is not like matured in the thymus. That's why we have very tiny thymus. And when we studied the different markers, you can see this bar graph. This is the CD4 T cells, CD8 T cells, double positive T cells, and double negative cells. You can see it is significantly lower in comparison with the control. However, I have mentioned that when the no signaling is disturbed, then T cells has potential to produce the B cells. And here is the, like, we got the result B220, that is a marker of B cells, and it is significantly increasing in comparison with the control, and that is a clear indication that no signaling is disturbed because of the mutation in the POFUT gene. And here also, does fringes affect the immune cell development? I mentioned that fringes cause the lunatic fringe cause the spondylocostal dystrophy, where we can see the human being with the problem in the ribs. So here also you can see the control, where the oak neck, ophicose, and clip neck beautifully decorated in the EGF of notch. And when we knock out these fringes, however, you can see here oak neck is here, ophicose is here, but the fringes, this repeat is not further elongated. And we found that this is a normal mice, and when we knock out this small tail, there is problem with the rib. And so you can see the differences, how these glycans are important for the body development also, and the immune cell development also. And here if we compare the development of T cells, which we found that double positive cells is decreasing, and the CD4, CD8 T cells are increasing in the thymus. And in the spleen, we got decrease in the T cells and then decrease in the CD8 T cells. Okay, and then what is take home message from this talk is glycans are important, and they play a very pivotal role in drug discovery. They are required for the cell-to-cell communication, because we see that it disturbs the notch signaling also, and they are also required for the interaction between the cells. Different O glycans are required to maintain the lymphopoiesis and myelopoiesis. We have clearly seen when we knock out the different mice model, we found that the immune cell lineages are totally disturbed. And these glycobiology actually a very rapidly evolving field with a sweet future, because there are lots of things has not been done yet, and it's required more time, more funding, more grants to study the brief and to study in deep of all these glycans role. So this is all about this talk, and welcome to the lab of Pamela Stanley, where you can see the beautiful lady. She's a Pamela Stanley, Actually, she is a very famous professor in the field of glycobiology. In the world, if any lab is using the knockouts of these glycans, mainly in the Chinese hamster ovary, she is delivering that in entire the globe. So everybody knows her, and she has done a lot in the field of glycobiology. If you have read about the essential of glycobiology, so she is the author in that and editor in that. And this is my lab mates. This is a picture of mine from the lake house of Pamela Stanley, where we were discussing about my future, what I will be doing after my postdoc with her lab. And this is my lab manager, Subha Sundaran. She's an American South Indian and working for Pamela Stanley since 40 years. And this is the beautiful new building view of Albert Einstein College of Medicine. If you allow me, then I think it's all, I have already taken more than 1 hour, I think. If you allow me, then I can quickly discuss about the herbal informatics, IUR informatics. So,
yeah, Ankit, you can continue.
Okay, thank you, ma'am. So that was the one part which I was discussing about the eminence of glycans. And now actually this is a very important, and mainly for the first year, second year and third year student, you can do this model in your lab in the Department of Biochemistry only. So herbal informatics, we developed this model in 2014. I was the key player when I was doing this model, and INMAS DRDO division of CBN and Defense developed this model actually. So what this model is, so if somebody will tell you, okay, find some drugs for the rheumatoid arthritis, so most of them will go and read the papers from the PubMed, and if they want to use it, they will say, okay, these doses are using, we can also use these kind of dose and test our new model. And this model is totally eradicating, totally like removing this approach. This is the well-established model, that is a process and development of new drug, new molecules, new moieties, that is actually based on the web-based resources. And this is the 5-step based model, 5-step approach model, starting from the step 1. If I want to do research in the rheumatoid arthritis, what I will do, I'll go in the Google, I will read lots of paper about the rheumatoid arthritis, and I will find out which factors actually responsible to enhance the progression of this disease, and how many factors are there. If I'll regulate these factors, it can be like, it can give the relief from this disease. So this is all about the first step. We'll go read the pathophysiology very carefully and find out the factors that are responsible for the progression of disease. Then we have step 2, that is bioactivity parameter. And you can see here I put AR plus VF, that is anti-rheumatic agents. What I will do, I'll go in the PubMed, because PubMed is the standard engine we are using for our scientific literature. So we'll put this bioactivity parameter, that is called as anti-rheumatic agent plus virulence factor inhibition. Virulence factor inhibition is the disease factor inhibition, which we inhibit and can achieve the drug target. So when we'll search in the PubMed, we will find some hits. Actually, the PubMed and the Google works in the priority indexing, because if you go and read Ankit Tanwar many times in your Google, so when you'll open, or if anyone will open in their computer Ankit Tanwar will be the first option, because it works on the priority indexing, who searched more. So we set the criteria here, okay, just read 20 papers only, because that is mostly readable paper. And on the basis of that, we can make the percentage relevance, and then we calculate the percentage relevance using the unique mathematical model, the simple mathematics. Then we got some relevance percentages here. Then in the third step, what we can do, if we are interested in herbals or the phytochemicals or any the chemotherapeutic moieties or anything, what we will do, we will go in the Google, we'll search and make a database of 50 to 100 herbal compounds or any agents that we want to use. we will describe them very carefully. We will write down about their all the medicinal properties and all, if somebody has used to target this disease or any factor which we have used in our model. After this step, we will go for the step 4, that is also the bioactivity parameter approach only. But you can see here I have mentioned the herbal and then virulence factor invasion. So what I have found in the third step, I'll test whether I have found the good molecules or not. So here we'll search whether in the Google search engine or the PubMed search engine, and we will search whether this herbal compound or the compound which we have selected in the step 3 has any potential to inhibit this virulence factor. So we'll find any paper. If we'll find one paper, then we'll give it 1 score, positive score. That means this plant is working for this disease which we are targeting. If we didn't find anything, we will just put 0. This is a totally null hypothesis, whether we will take it or that will be out. So we'll make, on the basis of that, we'll make the scoring. Then this scoring we will be calculated by the weightage score and optimization. And actually, if you want, if you are really interested, I can give you a detailed lecture in that also, because here I cannot explain the mathematical model here. Then after this 5th step, when we optimize the herbal compounds based on this scoring, we will get, like in the starting I have selected the 5200 herbal compounds, right? At this 5th step, we will get only the 5 or 6, and this is the beauty of this model. So we have selected the herbal compounds, but our model scrutinized only 5, and that will be easy for us to test on the molecular docking level. That we just need the ligands and their receptor, and we can dock them in the various softwares are available. Hexcuda nowadays, like Autodoc, Vina, and Schrodinger, that is a very famous and expensive tool. And we made the physiological function indicator based on their scoring. And then we have another tool. Okay, so this is the in silico approach only. So in the in silico approach, we can also identify whether this herbal compound has any toxic effect or not. So we have the software that is developed by the Environmental Protection Act, I think, agency, sorry, toxicity estimation software tool. And this software can tell you the lethal dose of that compound, lethal concentration, their bioavailability, their pharmacokinetic properties, everything this software will tell you on your computer. And on the basis of that, you can make the mole inspiration profile also to predict their bioactivity prediction, how much they have the ion channel modulation activity or any enzyme inhibition activity. So this is all about this model. And then if you've done this, so you can whether you can publish your paper on the basis of this model, or this model you can take it into an account to study further at in vitro and in vivo level, where you can use the cell culture or the animal model. Okay, and this is the recent accomplishment from the herbal informatics model which we have achieved, in which Professor Rasmivardhan has contributed 3 papers, and I think 3rd year batch of students also got name in these papers. And so they have done all the herbal informatics model, and now I think they are doing the molecular docking and other prediction analysis, and I think another 3 papers will be in pipeline. So, and then when I have developed this model in 2014, so I have given a talk in IIT in the front of our former health minister, Dr. Harshvardhan, where there is a conference going on, IISF, Indian International Science Festival, where I've talked about this model, and it was in limelight, and they have published my name also. So there was one reporter who wants to write about me and about this model, and they have written in 2015, and then it was in the various paper also. And this is my accomplishment, which I have, the work which I have done so far based on that. And I am very grateful to Shivaji College, because whatever I am doing today and whatever I am here, just its credit goes to all the highly skilled professor of our department. because they have trained me like that. Nowadays I can utilize my biochemistry with my toxicology research and my animal model research. And if anyone from you is really interested in herbal informatic model, we can discuss anytime and we can start working on it. Thank you so much. And now I'm open for any question if you have.
Thank you, Dr. Ankit. Thank you very much for such an informative talk. Now I think I hand over to Usha, Dr. Usha. She can continue for the questions.
Thank you, Dr. Ankit, for enlightening all of us about the importance of Glycans and hematopoiesis, and many congratulations to you for your achievements. So before we take the questions from the participants, I request everyone to put their video on